Well, everyone, I want to welcome you to tonight's lecture. I am very, very excited to be able to introduce our speaker tonight. Professor Bill Dennison is Vice President of Science Application at the University of Maryland Center for Environmental Science since 2002. Bill created the Integration and Application Network, which is a boundary organization comprised of scientists interested in solving, not just studying, environmental problems. He was involved for 10 years with the Healthy Waterways program in Queensland, Australia. Bill was trained as a marine scientist. He got his master's degree at the University of Alaska, went on to get his PhD at the University of Chicago, and began developing transdisciplinary science skills as a postdoc fellow here at Stony Brook University and as a faculty at the University of Queensland as well. Bill and his team of science integrators and science communicators have pioneered novel science communication techniques. They have co-produced environmental report cards around the world, including Chesapeake Bay, the Great Barrier Reef, Mississippi River. Bill is co-chair of the Environment and Society Foundation in the University of Maryland Environmental Graduate Program, integrating social and natural science. And he has just recently received the inaugural Margaret Davison Stewardship Award from the Coastal and Estuarine Research Federation. Tonight, I am very excited. Bill is going to be talking to us about tomorrow, about today's lecture, bloom busting in Great South Bay, Australia, and Chesapeake Bay, what we know and don't know about the ocean. Thank you so much, Bill.
Thanks, Brad. Great to be here, and I'm really excited to be able to tell this story tonight. Let's see if I can pop up the slide. Can you see that? Okay, yes. So I called it bloom busting in Great South Bay, Australia, and Chesapeake Bay. And I'm going to tell you 3 stories from this relatively long career that I've had in this world. The first story, I'll talk about discovering the brown tide in Great South Bay. The second story will be an Australian story I call the perfect slime. And the third will be the Chesapeake recovery story. So let me start with this first story. And the first story comes about because the director at what was called the Marine Science Research Center at the time in Stony Brook, Jerry Schubel, was able to go to the Donut King down in where were they, in Bayville or something, down in on Long Island, and convince them to create a postdoc, an Intimans postdoc, which they call the Coastal Marine Scholar. I kind of converted that to the Coasting Marine Scholar, but I was able to join the Marine Science Research Center with Carmela Cuomo, was the other Coastal Marine Scholar. And as you see here from the description that was written when I joined, was that I was going to be working with Doug Capone on the nitrogen cycle of seagrass sediments in Great South Bay, investigate the relationship between nitrogen availability and plant growth, and then determine how oxygen is transported to the leaves. That didn't happen. I'm going to tell you the story of what actually did happen, but that was the intent when I started at Stony Brook. So I had gone to the University of Chicago, 1,000 miles from the ocean. I had done my master's in Alaska, 450 miles away from the ocean in Fairbanks. So I got a job on the ocean. I was going to fully live on the ocean. So I bought an old 1955 wooden boat, called it Ador. That's my mother's name spelled backwards. She had died when I was in grad school. And that's my family that were on the fantail of the boat with me. And I loved living on that boat. I moored the boat in Corey Creek in Blue Point, which is due south of Stony Brook. Just take Nichols Road down south till you hit Great South Bay. My boat's on the left. My neighbors were ducks and geese. There's a clam boat to the right. And then for geographic reference, for anybody on Long Island, you probably have heard of Flo's. It was an iconic shack that did toasted sandwiches. I guess it closed this year because of COVID, but they were my neighbors. So what I did was I would go out and sample seagrass in Great South Bay to start this project that I had envisioned. But I couldn't see my hand in front of my face. The water was really brown. And I would come back and go to the lab and say, it's really turbid out there in Great South Bay. And my colleagues at the lab who had lived in Long Island, I just arrived, said, oh, well, it's shallow and it gets resuspended, it gets mixed up really easily, you know, a little wind. And so, well, it wasn't very windy. Well, when it rains, there's runoff and it brings in sediments and turbidity, and I said, it hadn't even rained. So I was starting to get a little confused about this. And so what I did is I brought a sample back in a jar, and Ed Carpenter, who was the phytoplankton ecologist, put it under a microscope, and all he could see were little brown dots. Couldn't identify what it was. He'd never seen it before. Just a whole lot of little brown dots. Well, as it turns out, then, as he's looking in the microscope, two things happen. A shellfish guy, Scott Siddall, came by and said, the scallop baymen out on the east end of Long Island are saying the scallops are dying. There's something going on. There's something in the water. And Linda Duguay, who was a faculty member, also picked up. She's from Rhode Island originally, and knows the people at University of Rhode Island really well. So she said, yeah, they were talking about it at Narragansett Bay, and the mussels were dying. So what I did is I hired a plane, and we flew around, found it in Narragansett Bay, Peconic Bays, Great South Bay, and Barnegat Bay in New Jersey. And then one of the scientists at the University of Rhode Island, John Seaberth, used electron microscopy and identified this as a species that was new to science. It had never been described before. And he called it Ariococcus, little brown round ball. And so that sort of launched us into a whole series of activities. Well, one of the things that was going on, as I indicated, was the scallops were being decimated out in the East End. They couldn't feed on this organism. They were starving, literally starving to death. Well, it turns out because of the scallop fishery, and it wasn't helping the clams, but the clams in Great South Bay were surviving, not like the scallops. But it made the New York Times. It became a big article, a big issue with the New York Times. They covered the story, and they coined two important terms: the brown tide and the Bloombusters. So I'll just read you this. It says, Dr. Dennison is part of a small group of scientists at Stony Brook devoting special attention to the brown tide. Informally, the researchers have been called Bloombusters, but that label is more optimistic than realistic. That was actually pretty prophetic. It was in fact optimistic. And I think Bill Wise was associate director at Seagrasses at Stony Brook, was the guy that came up with the Bloombusters. And we tried to talk him out of it, but the the media loved it. So we ended up Bloombusters. Well, we got lots of articles, and they come out on the boat with me, and I take them around, and I got in a little bit of trouble from quoting these things. I said, in one article, I said, we've all been pushed to the limits by this. We're realizing how little we know about the ocean. And then another article, they actually pulled out the subtitle, said, we know so little. We're still trying to figure out what's going on with the brown tide. We know so little about the ocean. Well, that's what got me in trouble, is I was talking about how stupid we were. So Ed Carpenter and Doug Capone, who were my postdoc mentors, they kind of come up and said, well, Bill, you may not know much about the ocean, but we sure do. So just keep that to yourself, okay? Keep your ignorance to yourself. And then it also affected my family. So I was fortunate to be quoted in the New York Times because up until that time, my grandmother back in Ohio, who ran a small town newspaper, had thought I was wasting most of my life, but I was in the New York Times. For her, that was the holy grail. The problem was what I was saying. And so she started saying, what have you been doing in school all this time? Haven't you learned anything? So I wasn't winning friends or impressing my colleagues or my family by being this way. But I think what it was is I was ahead of my time. 20 years later, in 2005, the ocean literacy principles were developed with a team of a couple hundred scientists. The first one was that the Earth is one big ocean with many features. Second was the ocean and life in the ocean shape the features of Earth. 3, oceans a major influence on weather and climate. 4, the ocean made Earth habitable. 5, the ocean supports a great diversity of life and ecosystems. 6, the ocean and humans are inextricably interconnected. And finally, number 7, this is mine, The ocean is largely unexplored. You could just say we know so little about the ocean. So I just think I was ahead of the time, that's all. Anyway, what Ed and I kidnapped Jerry Schubel, the director, and said, we need some help on this. We need somebody that knows something. So Liz Cosper was a research faculty member at the Stony Brook, and so we were able to get Liz funded and engaged in this project, and we reached out and worked with Bob Nuzzi out of the East End, East Hampton, and Liz put together a large international conference. And that conference generated a big fat book on novel phytoplankton blooms, causes and impacts of recurrent brown tides and other unusual blooms. The blooms started popping up in Laguna Madre, Texas as well. So we went down there and visited those guys. So it became quite a big research effort. So out of the book, I got 2 pages I scanned, and I'll show you a couple things that we learned. One, back to my seagrasses, which I came to Stony Brook to study. And what I was doing is documenting their demise because of the brown tide. It turns out seagrasses need a lot of light to survive. They can grow to the depth of about a Secchi disc, and a Secchi disc is a dinner plate, and it's named after the Italian who threw a dinner plate over the side of the boat. And so it's in fact, it's 30 centimeters, the size of a dinner plate, and you lower it down until you can't see it anymore. And that's about exactly where the grass can grow to. That's the depth that they get cut off because of the lack of light. Well, what happens with those little brown dots, the brown tide, was that it reflected a lot of light. And so the Secchi depth went to about half of its original depth. It used to be about 4 meters, it was 2 meters. If it was 1 meter, it was 2 meters, it went down to 1 meter. So on the right-hand side, what you see is a graph of the density of the eelgrass pre-bloom, the ones on the right, versus post-bloom, the ones on the left. And you can see that the depth was about halved, and of course the biomass was dramatically declined as well. So it really had a big impact. So the big question was, why is this bloom? Where does this come from? This thing brand new to science. What's going on? Where is it from? Well, we wrote a paper at this point. This is arm waving. We didn't have we had a lot of fragmented pieces of data, and we said, well, the period preceding the induction of the bloom was relatively dry, but then there were pulses of rain that would bring runoff and groundwater. The bloom was initiated inshore. It was in a period when the sea was fairly quiescent. There wasn't very much flushing with the ocean. The salinities got high, and there were a lot of organic nutrients. And whenever we fed it different nutrients, it really got stimulated with organic nutrients. So that's how we left it back, and this is 1987. So, and then of course I left Long Island, but what's gone on since then is one of Brad Peterson's colleagues, Chris Gobler at Stony Brook, has been very actively, and he was Liz Cosper's student, so continuing the legacy there, has been very actively looking at brown tide and other harmful algal blooms in Long Island. And this is a paper that he and a student published this year that shows that these things are on the rise globally. We're going to see more and more of these. I went and had a look just the other day at the Google Scholar citations for brown tide, and it kind of blew me away. There's 63,000 Google Scholar entries, and that's something that 35 years ago didn't even exist in the scientific literature. So it went from nothing to a lot relatively rapidly. So 3 big things happened to me at Stony Brook that changed the course of my life. One is I tagged along on a research trip that Doug Capone and Linda Duguay had organized to go to the Australian Institute of Marine Science. So I got to be introduced to Australia and Australians, and that planted a seed that eventually resulted in me going back there for 10 years. I also was captivated by the fact that my science was something people cared about and it was making a difference. The Bayman and Long Island in particular, and the climbers that were my neighbors in the marina were being affected. So I developed a lifelong interest in science applications. and I met a girl. This was one of Doug's graduate students, Judy O'Neill, and I married her, and 31 years later, we're together. And I dragged her down to Australia when I had an opportunity to go to the University of Queensland, and I was able to form a group, an active science group called the Marine Botany Group. We started out just doing strict sort of science of ecophysiology, looking at all the different plants. We had mangroves, corals, seagrasses, macroalgae, phytoplankton, etc. But then we kind of started developing this big cleanup campaign called Healthy Waterways. And so we started supporting that and set up monitoring programs, and actually started doing environmental report cards, which you'll see later in our Chesapeake story. And then we went to on to an Australian assessment with several groups to assess Australian estuaries. Had a great 10 years. It was a lot of fun. And so that's the setting for the story number 2, which is I call the perfect slime. Now, this is a chapter in a book that I co-wrote with Peter Oliver, who you're going to meet in a couple minutes. And we called it Dancing with Dugongs, Having Fun and Developing a Practical Philosophy for Environmental Teaching and Research. And dugongs are the cousins of manatees, and there are 1,000 of them in Moreton Bay, and 10,000 green sea turtles. So, and so when this Lyngbia, this bloom, this slime came along, the dugongs and the turtles were the ones that we were really worried about. So the story starts because fishermen were getting skin lesions, and you can see these were pretty nasty skin lesions, and there were rotting mats of this algae washing up on the beaches of Moreton Bay, so much so that they had to take backhoes and dump trucks to cart it off to the landfill because it was rotting and stinking. And in fact, I had a woman come down with her silver from her silver cabinet and saying, "This silver's tarnishing because of the rotting grass. What can we do? What can we do about it?" Well, and this is an interesting part of the story. The fishermen were experiencing it the previous year, but they weren't talking about it. This had a very severe contact dermatitis, as you saw from that guy's legs. And it turns out your hands are fairly immune to it, but not any of the rest of your body. The bottoms of your feet and your palms of your hand were pretty good, but the rest of you was affected. And these fishermen were not washing their hands after they handled their fishing gear and going to the bathroom. And so the first place that some of these skin lesions appeared were on very sensitive parts of their body. And they weren't talking about it because they thought they had a social disease. And so it took a while to break the ice, but Greg Savage kind of broke the ice, and he found the local catchment coordinator, Peter Oliver, the guy that wrote this book with me, and said, "There's this terrible thing happening and it's really bad, so can you help us out?" So Peter dutifully got a bag of this, a plastic bag of this blue-green algae. He didn't know what it was, just this slime. And he walked to the University of Queensland, and he didn't know anybody there. He just walked around and said, "Does anybody know what this is? Who can tell me what this is?" And then somebody eventually directed him to the marine botany lab, my lab. And Judy was there, and Judy had just finished her PhD with Doug Capone and Mike Roman and Ed Carpenter, and had been studying another marine cyanobacteria, sea sawdust, trichodesmium, out in the middle of the Atlantic Ocean. So she was able to quickly put it under the microscope, identify it as Lyngbia majuscula, and looked it up online, and it said, it had the sheath and had all these morphological features, but it also had a contact skin dermatitis. And then we got on to the Hawaiians, who called it stinging lemur, and they had an outbreak in the early 90s. So we figured out that this was the agent that was causing all the problems. So as scientists do, we started studying it. We grabbed some samples with the help of Greg Savage and Peter Oliver, and we started doing bioassays. We squirted different nutrients in there, and we found that what really took off was when we added iron. So we gave it a little shot of iron, and that really made it grow fast. And that's something that's these cyanobacteria are pretty famous for, is that they require a lot of iron. It has to do with their ability to produce energy, and with a chemical called ferredoxin, and to crack nitrogen molecules with an enzyme called nitrogenase, all of which needed a lot of iron. So they needed a lot of iron to grow. So we went out and looked for iron. We got to find the iron. So we chased out, and we looked for iron. We found a couple streams that were orange, that were precipitating iron, and we thought, wow, we've on to something here. And it was right next to a canal estate, and then that became oh, maybe it's this canal estate, because they've recently constructed it. They dredged out the sea bottom, which has a lot of bound iron in it, and then lift it up and put it on land, and that could actually create an opportunity for iron to run out and leach out into the ocean. And then about that time, we got a call from Max Haste, the head ranger up at Fraser Island. This stuff was on the media, so he had heard about it, and he said, "Oh, I got some stories. We've got campers that come in. This is a national park. These campers are coming in with these lesions on their skin, and one of the ranger's boys stuck his head out the window as they're driving up the beach, and his whole face, including his eyelids, were peeling off. He said, it's really nasty. Can you come up here? Can you help us?" Well, so Fraser Island is about 2-hour drive north of Brisbane, where University of Queensland is based. And it's the largest sand island in the world. It's a gigantic sand island. And you drive on the beach 60 miles an hour for a couple hours to get to the ranger station after you take a ferry across. So we did that and got up there. And at the time, our two girls it was school holiday. So I thought, well, let's make it a family trip, and we can bring the girls up and go camping. We'd always wanted to get to Fraser Island. We hadn't been there yet, so we wanted to go there. And I asked Max, I said, can I bring the girls? And he said, no, you're not going to want to do that. I said, well, why not? I said, because the dingos are nasty. Well, it turns out Fraser Island has the largest population of purebred dingos in all of Australia. These are Australia's all marsupials, except for some bats and these dogs, the wild dogs that came over with Aboriginal Australians 40,000 years ago with the land bridge. And they are wild dogs. They are not domesticated whatsoever. But they also, on Fraser Island, had these wild horses, kind of like Assateague Island here in Maryland. And these wild horses, they call them brumbies there, were obviously exotic animals. And so they culled them. They thought that this is a national park. We want to keep it the way it should be. So they got rid of the horses. Well, it turns out the dingoes were feeding on the horses, or the babies and the old sick and dying horses. So they got very hungry. And that meant that they started getting very bothersome to the people visiting. And it resulted in the second case of a dingo killing a 9-year-old boy that tragically on Fraser. And then Max and his other rangers had to go out and do a cull on the dingoes, because the dingoes had gotten too aggressive. And it harkens back to a story that you might have heard about, where this couple, Lindy and Michael Chamberlain, had a little girl when they're on a camping trip to Uluru, or Ayers Rock in the photo at the top here. And their girl was taken from a tent, and they never found the body, not to this day. And they actually arrested the mom and put her in jail for several years, because they didn't believe a dingo could actually eat a baby. But Meryl Streep played her in the Lindy Chamberlain in the movie, and that's famous line, "The dingo ate my baby," came from that. So the dingoes were a big issue, and then the stinging lemur, the fireweed, was also an issue. So the rangers were having a hell of a time. So we braved the dingoes, went up there with just a graduate student. They were chomping around as we were launching the boat and everything. They were definitely around. And we went diving and found some lingua growing on these outcrops of this rock that they colloquially called coffee rock. There's a picture of an intertidal coffee rock outcrop. So we put together this diagram that kind of gave an idea of what we thought was going on. It didn't turn out to be quite correct, but this is what we thought at the time. We thought the canal estates on the left were being, where they took that sediment from the seafloor and put it up on the land, And when you aerate marine sediments, what happens is it converts the ferric sulfides into sulfuric acid. And the sulfuric acid leaches metals out as it runs back into the sea. And then these concretions of iron and aluminum, as they call it there, would be formed. And so that was the source of iron that we surmised. We also looked at the coffee rock, and we saw that that's formed by slightly acidic rainwater that's percolating through this very sandy soil. And then when it hits the water table, it turns into this concretion, which has a lot of iron and organic material. So that's what we thought was going on. We had a couple students get involved. So Judy partnered with Greg Savage to track the bloom, and Andrew Watkinson went out with Greg, and they mapped this bloom in 2000. And it turns out this bloom expansion rate was equivalent to 100 square meters a minute for a 2-month period, almost 2 months. So it's a really rapidly growing and spreading, and it made mats that were at least a foot thick, if and sometimes more. But as we were out there sampling, we saw that there were occasions when the tides would turn and the wind would blow the right way, where this very dark stained water would come out across the site where the seagrass bed, which was covered by Lingbyia. So we were literally out in the boat when we saw this the first time, and we gave our one of the lab techs a cell phone and a credit card, and ran ashore and said, call a cab, get to the airport, get a plane, and fly around, and let's track where this thing is. And meanwhile, on the boat, we went and sampled, and you can see where we sampled. And we did some driving and sampling. And we sampled these places, and we found that there was a huge amount of this humic stuff. And it was, I mean, it was not subtle. You can see the picture of that's not concentrated. That's the raw water was very tannin stained. Well, when you got in the air and we looked at the photos, we realized something was going on. And what was happening, they had the previous year, they'd had a bushfire, a gigantic, well, this is actually 3 years prior, a gigantic bushfire. And this fire, and you've heard about it in the news last year, Australia was on fire about a year ago. This was resulted in a massive burn. Now, what you're seeing are in the red and pink are not actual fire or burn areas. Those are areas that were deforested, that were logged. The fire went through and destroyed the tree growth. So it was there's a lot of wood standing. So what they had to do to salvage that is cut it all down. So they went through and did a massive deforestation. It was very unusual. That's not the way they normally manage this forest. These are exotic pines that come from the US that they grow in Australia for fiber, for wood and timber, for building supplies. So, and then the other thing you can see from the photo on the right is that there was standing water. What was happening was as removing the trees, there wasn't any evapotranspiration. The needles weren't sucking the water out of the roots and evaporating it out into the atmosphere. So the groundwater came up to the surface, and all of a sudden the surface was inundated, even though it hadn't rained. So you can see the furrows there of the inundation. So we had another graduate student, Simon Albert, go and collect some sediments from all around the different potential sources, and put some Lingbyia in there, and then add this extract. And what we found from that was that the coffee rock and the canal development didn't do anything for it. We were on the wrong track. But what we did find was the pine needles, the pine plantation, either the intact pine or the cleared pine was causing this to bloom. And it was really great we did all kinds of photosynthesis and nitrogen fixation measurements, but you could literally see in the containers when we added the humic stuff, the Lingbyia would start to fizz because it was photosynthesizing so much, and it would rise up into the beaker. So it was a pretty clear demonstration that was stimulating the bloom. So we had to figure out how the iron and humics were related. So what we did is we recruited some scientists from the University of New South Wales down in Sydney to come up to Brisbane and help us. And so David Wayton and several of his students, including Andrew Rose, came up and did some really sophisticated iron chemistry measurements. David had trained at MIT with a renowned person in marine biogeochemistry. So what he found in his students was that this is a complicated, and this is the simplest version I could come up with. Those guys have very complicated science communication. But what was happening was the iron was binding in a complex with the humics, with the pine extract. And then with ultraviolet light, that complex would disassociate, and then the iron would be bioavailable in the seawater, which normally would precipitate out, like that photo of the stream with the orange stream. Normally it just precipitates out. So this was a big finding. So then we had a mechanism. We put the story together, and this is how we think the Lyngbya thing actually works in the end. The groundwater was raised because we did the mass deforestation, cutting off evapotranspiration. It allowed the iron to become in a more reduced stage, which is the bioavailable state, and then complex with the humics, these humic compounds complex, and then run off into the sea and then into that area where the Lyngbya started. The ultraviolet light would then break that complex and make that available for the Lyngbya. So that was the story we were able to eventually, and that's a whole bunch of scientific papers and theses that went into that, but that's the quick version. So we were pretty proud of ourselves, but then we got another phone call, and this time it was from a helicopter pilot on the Great Barrier Reef. So there's this weird little reef up in Hardy Reef, up on the Great Barrier Reef, that has this unique little micro atoll that looks like a heart. It's like the size, it's like a kiddie swimming pool. It's about 3 feet deep and about as big as your little kiddie pool. And it was very popular for tourists. So they would helicopter them out to this helicopter platform, and they'd take a little tinny out, a little boat out, and put them in there and get some photos, and that put them into their honeymoon album. So it was very popular. Well, this helicopter pilot said, I've been doing this for 27 years. This is the first time I've seen this, but there are these blooms going on. Can you come and have a look at them? So Judy went up with Andrew Watkinson, and he flew them out there. And lo and behold, on that platform, as they were landing, there were hundreds of seabirds, gannets and boobies and cormorants that were perching on the platform, because it's the only raised bit anywhere to be seen in the Great Barrier Reef. Everything else is underwater, and it was full of guano. The fish had the seabirds had been feeding on the fish, and then accumulating all this guano as they were pooping on this platform. So they scraped off some guano. We brought it back, and sure enough, the Lyngbya took off. So we found another source of these blooms. So kind of in retrospect, what we did is we created a monitoring and management program. We mapped it. It is a regular mapping program that occurs. They developed ways to suck it out of the mangroves and off the beaches to kind of alleviate the impacts of the bloom. Still going on. The LA Times got wind of the story, and Ken Weiss won a Pulitzer Prize for a five-part series on Lyngbya. And Greg Savage featured in there. You can see there's a little video clip that's online if you Google Altered Oceans. Ken wrote, and there are a lot. He had stories of red tides and everything else around the world, brown tides, but this was the feature story. And he said the fireweed began each spring as tufts of hairy growth spread across the seafloor fast enough to cover a football field in an hour. When fishermen touched it, their skin broke out in searing welts. Their lips blistered and peeled. Their eyes burned and swelled shut. Water that splashed from their nets spread the inflammation to their legs and torsos, and he left out the other bit that I had told you earlier, the other bits that were also affected. So check it out if it's really interesting. I did the same thing as I had done for the brown tide. I went to Google Scholar, and sure enough, not 63,000, but 2,600 Google Scholar entries. So again, it's a pretty prolific research area. So that's the second story. Now, the third story started when I returned to the US and set up this integration and application network that Brad mentioned in the introduction. So what we did at the University of Queensland Center for Environmental Science is we redefined scholarship. It's usually research, teaching, and service, typically in that order in terms of priority. But in this case, it's equally weighted between discovery, integration, application, and teaching. That's the origin of the term integration application network, comes from that definition of scholarship. So we're trying to make science relevant. So this is the story about Chesapeake Bay. Well, Chesapeake is very large, but very shallow. Its average depth is only about 24 feet. And it has a gigantic watershed. It extends up to Cooperstown, New York. It includes half of Pennsylvania, half of Virginia, all of Maryland, District of Columbia, bits of Delaware, and West Virginia. So that result of that big watershed and that shallow bay makes it really productive. It's got some of the most amazing shellfish and finfish productivity of anywhere in the world, but it's also very vulnerable as a result of that productivity. So that's the setting. The algal blooms have been impacting Chesapeake Bay, and you can see all kinds of bad characters have been infecting all the waters of Chesapeake Bay, different types in different places, causing fish kills. And even look in the Maryland coastal bays here, Chincoteague Bay, a brown tide appeared in 1993. That's not part of Chesapeake Bay, but I just want to point out there's a link to the brown tide. So these algal blooms create oxygen dead zones as the phytoplankton sink and down to the bottom and are respired broken down by bacteria. And you can see on the left, the quote dead zone, the low oxygen area, and that's in the old river channel of the Susquehanna River that forms the center part of the bay. And every summer there's a dead zone. In this case, this is the worst one in 2011. And that dead zone has all kinds of implications for, of course, fish and shellfish can't survive. It creates differences in the organic matter processing and can be a real problem. So as a result of the dead zone, Chesapeake Bay has had a series of agreements that started in 1984 for a voluntary nutrient reductions. And generally they were talking about 40% reduction. But they didn't really get very far. It was only until 2010 when a regulatory program called the TMDL, Total Maximum Daily Load, was instituted. We call it the nutrient diet for short. And we have quantifiable 2-year milestones for reductions of nitrogen and phosphorus and sediments as a result of this TMDL. And now we're starting to see some progress. So this is a photograph of Blue Plains Sewage Treatment Plant, the largest advanced wastewater treatment plant in the East Coast of the United States that takes wastewater and reduces the nitrogen phosphorus to the sort of state of the art. Really good job. And where we see Blue Plains and some other sewage treatment outfalls that have been improved in terms of releasing nutrients, we see that the resurgence of our seagrasses. So this is the good news for especially somebody who's been going around the world to study seagrasses and finding these blooms killing it. The other thing that's gained us some benefit and benefiting Long Island is that we're downwind of the airshed, which is much larger than the watershed, extends all the way into the Ohio Valley. And that means that the catalytic converters on the tailpipes of our cars and the smokestack scrubbers on the power plants have reduced NOx, and that's resulted in lower stream nitrate. So we're getting some benefit of the air quality improvements. So as a result, we started to see some really exciting things happen. At the head of the bay, at the mouth of the Susquehanna River, we see, and this is a Google Earth image I just downloaded, you can see the bed from Google Earth. I mean, this is big enough for the satellite picks it up. And there's a picture on the right of some guys standing in this knee-deep, thigh-deep water where this seagrass bed is occurring. And here's a little video of that bed. It looks like Florida Bay. It's brilliant, clear water, fish and crabs all around. And in fact, there's tufts. If you look and see, among the seagrass, some of the dark tufts is actually another species of Lingbia that's growing in there. And you can see that the area doubled, but more impressively, the density really expanded. Up until 2011, when the second biggest runoff event in the recorded history of Chesapeake Bay occurred, associated with Tropical Storm Lee, and knocked it back, but didn't knock it out. And so the good news is, and this is only dated to 2015, but it continues to expand. So it's definitely on the mend. We put together a team of senior experienced old people with a bunch of young, smart people. And we did a series of synthesis of the 35 years of water quality data and seagrass measurements from aerial photography. And we put together a series of papers. We published one in Bioscience, one in the Proceedings of the National Academy of Science. And we correlated, not just correlated, we causally linked the nutrient reduction to the abundance of seagrass. Now we call it in Chesapeake Bay submerged aquatic vegetation as an homage to the fact that it's not just marine seagrasses, but also freshwater and estuarine species. So this was well received because we were able to get it into the Washington Post, our local newspaper, right when the Trump administration was trying to cut the funding of the Chesapeake Bay program. So it turns out we've got funded, in fact, got plussed up, and funding is actually record levels right now for the Chesapeake Bay program. We also put it out in this journalistic blog called The Conversation, and it got picked up by San Francisco, LA Times, San Francisco Chronicle. So it was great to get a large media coverage of this because it really did help us. So in the end, we can say that the bay is still degraded. Oh, actually, I'm gonna wait a second. While I'm here, I'm gonna show you a little video that that I produced as a result of this this story. So here it goes. Yeah, I thought you guys in New York would appreciate this.
Start spreading the news. We're talking today. I want to be a part of it. Chesapeake, Chesapeake, your aquatic grasses, they are rebounding in the bay, and they are the heart of it. Chesapeake Bay, Chesapeake Bay. I want to wake up on a bay where the grasses grow deep and find your bay getting better, top of the heap. Your Chesapeake blues, they're melting away. Let's make a brand new start of it in Chesapeake Bay. If you can make it there, you can make it anywhere. It's up to you, Chesapeake, Chesapeake.
All right, there's a little more, but in the interest of time, I'm going to keep going because I have another song coming up that I want to share with you. This one is, get rid of this. Oh, it just wants to keep going. I gotta get rid of it. Make it go away. All right. That's on YouTube. So if you want to watch and sing along later, you're certainly welcome to do that. Now, back to the Chesapeake story. We are now seeing our report card results are that we're still degraded. We still get a C-, but the important thing is we're not degrading. We're actually turned the corner, and in most places it's getting better. This was last year's report card. It wasn't a very good year, but this year has been great. The grasses are booming, and we've got the second smallest dead zone just came out last week on record. And then another great story. We have a colleague, one of our faculty at the Chesapeake Biological Lab, was preparing to go to the Atlantic Ocean and put out some acoustics to look for marine mammal activity in anticipation of a wind farm out there. So she put these acoustic hydrophones off the dock and heard all these dolphins in Chesapeake Bay, and created an app, and now there's reports of hundreds of dolphins that have come back into the bay. So that's pretty exciting. So what have we learned from these three stories? Well, I learned that we actually do know something about the ocean, but I still will attest that there's many things we don't know. We can see from our Chesapeake example that volunteer efforts are really great, they're important, but we do need regulations to really make real progress in restoration. It's a Clean Air Act, the Clean Water Act, and the Chesapeake Agreement, that TMDL, that's really made a difference. We can see that our ecosystem degradation is reversible, the ocean is resilient. What I learned personally on this was that life is what happens to you while you're busy making other plans. So I didn't end up doing my postdoc at Stony Brook what I anticipated. I didn't do what I anticipated at the University of Queensland, but it's all good. So that's my lesson. So I wanted to play you a song designed just for this talk that was stimulated from the Bloombusters comment the New York Times made. So remember the time. So this bloom was in 1985. But in 1984, the #1 movie was Ghostbusters, and the #1 song was Ghostbusters. So I have a neighbor down here in Maryland. That student that was singing Chesapeake, Chesapeake, unfortunately graduated, and we let her get away. But I have a neighbor who's down the street, and she sang this song. Her name's Kat Parsons. And so Roy Parker Jr. did the Ghostbusters song in 1984. But Kat Parsons did this last weekend in 2020. We call it Bloombusters.
Bloombusters! If it's something strange in your neighborhood, who you gonna call? Bloombusters! If it's something weird and it don't look good, who you gonna call? Bloombusters! I ain't afraid of no bloom. I ain't afraid of no bloom. Seeing things out in Great South Bay, who can you call? Bloombusters! An invisible algae killing all your scallops, who can you call? Bloombusters! I ain't afraid of no bloom. I ain't afraid of no bloom. Who you gonna call? Bloombusters! If you're all alone, pick up the phone and call Bloombusters! I ain't afraid of no bloom. I ain't afraid of no bloom. I ain't afraid of no bloom.
So Kat came around with her little kids for Halloween last week, and the little girl was singing Bloombusters. She had heard her mom recording it, so I thought that was a nice touch. So I'd like to thank Brad Peterson in particular for inviting me, my colleagues from my wonderful time at Stony Brook University, my colleagues at University of Queensland, and my current colleagues at University of Maryland Center for Environmental Science. I'd like to give a shout out to Larry Swanson, who I got to overlap with briefly as a postdoc, but then served on committees. And Larry was a class act, and it was a real loss to Stony Brook to lose such a great scholar. So that's it for me. Thank you. Now you got that song going in your head, right, Brad?
Yeah, I do. I absolutely do. So I imagine that there are some on the call here who would like to ask a question of Bill. If you want to unmute yourself, otherwise we'll be monitoring the chat to see whether or not you want to would feel more comfortable typing your question in.
I have a question. Rich Grove from the town of Babylon. What species of aquatic vegetation rebounded at the head of the Chesapeake Bay?
Good question. There are about 12 species in that mixed species bed, but the big one was wild celery, Vallisneria americana, which is a really look-alike for eelgrass. It's got the same kind of blade morphology in it. It's like the freshwater cousin of Zostra.
Thank you.
Hey, Bill, this is Malcolm and Waveney Bowman. I just want to say hello. We're kind of marooned down here in New Zealand right now, but remember fondly the days.
Well, what a terrible place with zero COVID. You've done well, Malcolm.
But I remember the days we visited you in Adelaide, Australia. Brisbane, Australia, and you follow us back to the US. So it's great to have you back, and thank you so much for all the great work you and Judy have done over the decades that we've known you.
Thanks, Malcolm. And I know I read your tribute to Larry Swanson. That was really well done.
Oh, very sad.
Thank you.
Yeah, very sad. Yeah.
So, Bill, there was a question in the chat, and it was really talking about, kind of a report card for local waters. But I think the issue is kind of your development of water quality report cards and how useful they've been to the different areas. I know that the Peconic Estuary program right now is working on developing a report card for the Peconic Bay, and I'm just curious your perspective given your background.
Yeah, that's a good question. And it's really, report cards, we started it in Australia back in 1998. We were hosting the first International River Symposium. And one of the organizers of the symposium said, well, it'd be really good if you could just kind of simplify all the science you guys do, simplify it, just give us ABC, give us a report card. So, okay, sure, we'll do that. And literally, this is back in the day, we had it on a transparency. And so I was writing them in. And I just put half a dozen grad students together, and we kind of just said, well, we'll give that an A and we'll give that an F and everything in between. So we just made it up. It was just totally out of the air. Well, it turns out in this whole symposium, we had the first river prize. We had a delegation from the Mersey River in UK come, and we wanted to celebrate their great cleanup success. And that got on page 17. What was front page? Brisbane River gets a D. And we're like, well, maybe we better we're scientists, maybe we better do that with real science. So we developed a monitoring program and created a much more robust report card. And it became quite a thing. I mean, they're still doing it 20 years. They just released it yesterday, their 20-year anniversary of the Healthy Waterways. Now they call it Healthy Land and Water. So it's still going strong. So when it came to Chesapeake, so we got to do a report card, and we'd started that in 2007 and been doing it every year since. And in the last few years, the Chesapeake report card has reached, using social and traditional media, between 70 and 150 million people, way more than the 20 million in the watershed. So clearly the message resonates, and it really helps put attention and focus on what needs to happen to clean it up. Politicians actually like it if they have pre-warning, if they know it's coming and can respond. And so it's a really good way to reach a broad swath of the community and get the message out. And it's transparency and accountability. So we've now done report cards on every continent except Antarctica, and we've developed training materials. We have a book of how to do report cards. This one, sorry, that one's we did one in Mongolia, and they translate it to Mongolian, but we've got English versions, and it's available for free online. And we've got some training modules, and we're developing a course for edX, which is a global, massive open online course, MOOC. It'll be, report card class will be released course will be released in mid-February. We're trying to share it. We're trying to give all the tools and techniques and what we've learned to everybody that wants to do that, because we think it's a really valuable environmental management tool. We have a governor, Martin O'Malley, former governor of Maryland, who wrote a book called Smarter Government. And he created a thing called Baystat, which is really very insightful about how to manage, how to govern based on science. It's very exciting. So it's available on the Integration and Application Network website. If you Google my name and Google Maryland, you'll find your way to the website, and it's available on this thing we call Ian Press. And the Bay is very, Lingbia, interesting. We were up on Cape Cod in Provincetown for the last couple summers and found Lingbia growing there in Cape Cod. And I think Judy got a sample from somebody from Maine as well. So it does seem to be up in the Northeast. Are there scallops in Maryland? No, no scallops. Interesting project going on in Virginia. So the Delmarva Peninsula, Delaware, Maryland, Virginia, has a long skinny bit, and the seaside bays, the equivalent of Great South Bay, is in our case is Chesapeake Bay, but just south of that in the Virginia coastal waters, they have a large seagrass restoration project, which now they're seeding with scallops. And I think they had a GoFundMe site to buy more scallop seed. So there's we'll see how that works. We have had no luck in Chesapeake Bay. When we do the clam surveys, if you find a scallop, it's a cause of celebration. So no scallops. There's a great question about nutrients reduced too much, and I don't know about Mumford Cove. We did do the Long Island Sound report card. They're not doing with too little nutrients. It's still too many. But we're starting to see our Maryland forest are starting to become starved for nitrogen because we've reduced the air deposition, and with climate change, the growing season has increased. And so now we're starting to see, particularly toward the end of the season, the trees are becoming very nutrient limited. So I think the problem we're going to have, and this is true for nitrogen and phosphorus, we're going to have areas that are too much, and it has to do with animal manure that's very rich in phosphorus, and fertilizer that's made out of atmospheric cracking the nitrogen molecule. Is going to be places with too much and places with too little. So I think we're seeing in our future places that are going to be nutrient deserts and places that are going to be nutrient saturation. So we're going to have to figure out how to move the nutrients from the places where they're causing the pollution to the places that need it.
Hey, Bill, just wondering how, you got some grasses to respond in the Chesapeake to really big nutrient load reductions. And, there's a whole bunch of different species, as you go through the tidal freshwater, there's a whole bunch of big range to choose from there. We don't really have those riverine type systems here. So how do we prepare for the day when we do get our nutrient loads under control for what could be next, right? So there's not a million brant flying up the coast or down the coast every season, moving seeds around. People get frustrated with us when we talk about that. But how do we prepare for what could be next, right? We did this with corals, where we took corals in warmer water, we propagated them, we put them out in stressed areas, they responded. That's some real small efforts, but making a big impact. How do we think about that with grasses?
Well, my thought is that we have to rethink our entire strategy on restoration. Normally what we do is we maintain genetic integrity. We don't want new genes in. We may be thinking about trying to increase our genetic diversity and maybe assisted migration. Maybe we should be bringing, because one of our seagrasses, the same one you have, Zostra, is starting to burn out in the summer. We had a burnout in 05, a burnout in 2010, and that's fine because seedlings could come back the next year. But we had 2 successive burnouts in 2018 and 2019. Nothing coming back this year for that grass. So where the other stuff's going gangbusters. So it's going to be seed limited. Well, it's just on near the southern edge of its distribution. We might have to bring in halogely or another species from North Carolina and leapfrog it into the bay. Maybe we're going to have to just rethink how we do management. We're not looking for status quo. We can't be managing for today. We have to be managing for resilience for tomorrow. And as climate change accelerates, no matter what we do in terms of carbon emissions, we're still seeing these accelerating impacts. We're going to have to be really creative and think differently about it. We're going to have to manage for resilience instead of condition, and we're going to have to think very differently. So I would suggest maybe it's time to think about bringing some extra species in, bring some seeds in.
Thank you. Always good to
Well,
hear you, and
send
thanks for
them your
that
way.
response and tribute.
A question on the Delmarva, south of Chincoteague on the Chesapeake Bay and also the seaside bays. It's relatively undeveloped. Has there been any blooms in those areas, both on the seaside and the bayside?
Not really. That's really the most intact area. I got to tell you a quick story about the seaside bays. It's the reason they're undeveloped. And it's if you look on a satellite night sky, it's like the only dark bit on the whole sea coast of the Atlantic coast. And the Nature Conservancy came in and offered to buy up the land and the islands. And the locals said, no, we want to be like Ocean City. We want a casino. We want hotels. We're poor down here. We want to pump the economy up. So they said, no, we're not going to sell it to you. So what they did is they went away and sent down teams of lawyers, and most of the TNC leadership were lawyers, and they fly in a private jet, wear 3-piece suits, hire limos, go in there and say, I can't tell you what this is for, but we're interested in buying this up. Never lied, never told them what they were doing. Well, it turns out they were TNC, and they bought up the whole string of islands, and now you can get a true wilderness experience out there if you can tolerate the ticks and the mosquitoes. It's an amazing place, and that's where that great seagrass, the largest seagrass restoration in the world, is in those Virginia seaside bays. It started with one of my colleagues at Virginia Institute of Marine Science, Bob Orth, would go out there with seagrass seeds and just throw them out the boat. And the first summer he did it, the boat went in a straight line, and you can see this line of seagrass growing. And on the way back, he said, well, make some bends. And it turned out it was the letter B. And then the next year he went out, and it turned out he made the letter W. And then he got so enamored with spelling words that he got funding. He was able to get funded from NOAA, National Oceanographic Atmospheric Administration. So he spelled out NOAA. You can see it from the air. So anyway, that now is coalesced. You can't read the letters anymore. It's coalesced into a gigantic bed that they're trying to get the bay sculpts back in. And it's an amazing success story. And there's a large, long-term ecological research, LTER, site that University of Virginia runs. That's got a whole legion of students and researchers looking at that restored bed. It's a great case study. Yeah, I agree. Eastern Shore is an accident of geography, but I live on the Eastern Shore, and I think it's God's country. I love it here. It's a wonderful place. It's Long Island 40 years ago, when you still had the potato farms and it wasn't all developed. I shouldn't tell you guys, I don't want to populate the Eastern Shore with the 8 million people from Long Island, but it is a beautiful place. I'm going to have to check out Mumford Cove. I don't know that story. Thanks, Roger. I will check that out. Barry Truitt. He's the real deal. He's the guy who was behind that TNC buyback. There's a good book. I wonder if I have a copy. Actually, Linda knows this guy. Nelson Marshall was a longtime University of Rhode Island fellow, and he wrote a book called The Scallop Estuary about and it wasn't Mumford Cove particularly. It was what river is that? The whatever the Milford Lab is on.
No.
You can probably find it on Amazon. The Scallop Estuary by Nelson Marshall.
Is it the Connecticut River? I'm trying to remember. Was it the Connecticut River?
No.
No.
I think it started with the N. I should know this. I'm spacing
Yeah.
it.
True.
I want to introduce Doug and Linda. They were very important in my life. They're the ones that introduced. That's how I met Judy. That's how I got to Australia. They're the cause of a lot of my good times. We've had very good.
And you were
times.
never my postdoc.
And that's right. I was a post-degree scholar. I was the Intamin postdoc, not a Capone postdoc.
Hi, Malcolm. and Waverly.
Oh, hi, you guys. Hi, Doug. Hi, Linda.
You too.
Waveney's here as well, down under the ultimate deep south, you might say.
We were there ourselves
We were
for
in
a
Hamilton.
spell.
We were in Hamilton for 4 months.
You were? When was that?
November to February. We left just as COVID was coming to the United States.
Oh, really? We well, I don't want to take your time up, but
Yeah.
we came in March just as they were shutting down. New York was aflame with everybody getting sick. We've seen Jerry Schubel a couple of times out at the Query in the Pacific. He finally retired, I think, last summer. I don't know whether you've kept in touch with him.
Yeah,
Oh, yes.
I have. Well, Doug and Linda, big time, but I still run into them. I'm going to look them up. This
Oh, you should.
Going down memory lane. I gotta show them this talk.
I guess I was out there a year or so ago. I can't quite remember, but he looked great. I mean,
Yeah.
Oh, he's a great role model. He doesn't age at all.
So we were telling
And now
stories for his retirement. His, I guess, assistant
His assistant, yeah.
solicited stories. And I told an interesting story about an April Fool's joke that I was once pulled.
well, you gonna tell us?
Well, I'll tell a story because I think it's so. Ed Carpenter and Doug were really hot on the fact that they thought that they were underpaid. The salaries weren't equivalent to matching, and they got some salary data and they made little spreadsheets, Lotus 1, 2, 3 back in the day, and they made these comparisons, and they were really hot under the collar about it. So somebody wrote this memo back in the day when you had physical memos, and it said, look, I understand you've been working on these salary things during company time. I'm going to dock your pay accordingly. And I mean, I thought that was pretty outrageous. There's no way they're going to believe that. So they're out in the hall, and I could see them going back and forth, getting madder and madder. And they stomped off, and I was following and said, "Hey guys, what's the date on that? What was the date? April 1st? Is that the 1st of April?" And they didn't. And they were at Beeline, and normally I figured I had plenty of time because Jerry had sort of an inner sanctum, like 2 offices to get through. He wasn't very available right away. So I thought I could intercept him. Jerry happened to walk out right in front, and boom, and I just turned around and walked away.
Gosh, the closest I came to losing my job was I organized a regional conference at Stony Brook, and on the banquet at the end, I'll probably get killed for telling you this, but I organized a belly dancer to entertain the guests. And she was very gracious. I mean, she was a woman probably in her late 30s. Anyway, it's an art form. And after dinner there, this restaurant called the Bridge to Nowhere, Jerry Schubel was there, and Don Pritchard was there, and Harry Carter was there. And then this woman, she started wafting past these guys, and her veil kind of went over their faces, and she kept dancing, and then they left. The three guys left. I thought, oops, there goes my job.
Well,
How's Ed Carpenter doing? Haven't heard from him.
since we're telling stories, I got to tell the Bloombusters story. So Liz Cosper's husband, late husband, Terry, pretty wild man, was really into the Bloombusters. So he bought t-shirts, Bloombuster t-shirts for Ed and Liz and me, and had a party at his house. Were you there, Doug and Linda? It was.
That one.
No, I think we might have missed that.
You missed that one because at the party showed up two young women.
Oh, yes, we were.
With the boombox.
Yeah.
And they put on the Ghostbusters song and they proceeded to take their clothes off. Not all, the clothes, but lots of clothes were disappeared, a lot of skin. I just turned red. It was the most embarrassing
David
thing
Cameron
in
was
my
down.
entire life.
I don't
So I
remember
kept my clothes on for my version of Bloombusters.
Thank you.
that. So is Ed Carpenter still working?
Still
Yeah.
going strong, Malcolm. Yep.
Wow. You know, he's the only person I ever met in all my career who went from a full professor down to assistant professor. Gave
He
up his-
passed way fast.
Happily so.
He finally
gave-
went
gave-
back to
yes,
being-
he gave up his tenure and then zoomed back up again.
Yeah,
That took some balls to do that.
that's it. He's good. Still
And how
living
are
up
your
his
kids
hustle,
doing?
though.
What's that?
How are your children doing?
Our children are fine.
Very good.
They're all nearby,
Very close.
Oh, really? That's
Listen.
great.
along with 5 grandchildren.
Oh, my.
So Bill has administrative rights. And so I think you guys should stay on and spend some time with Bill. And I'm going to cut everybody else loose from this. Let me just, first of all, thank you, Bill, very, very much for giving this talk. This was great. Really appreciate it. I want to tell everybody before you leave that the speaker on Wednesday, December 2nd is going to be Matt Adjaman. He's from FAU. He's going to talk about cow nose rays in Long Island or in general. So thank you, Bill. Those who are ready to cut loose, you can cut loose. Please, Bill, stay and chat for a little bit. All right.
Great. Thanks, guys.
Thanks for joining us.
Thanks, Brad, for inviting him. It's been fun.